Hola, and welcome to the series where me, a fourth year med student, will be revising every subject. So, today we have first year, first topic embryology. And let's start. It all happens with fertilization. On day one, let me just quickly draw this out. And you can see this is a fimbriae and this is ampulla, which is a junction of fertilization. Actually, fertilization happens in ampullary isthmic junction. So on day one, this is a fertilized egg, which is also called zygote, and it undergoes cleavage or divisions to form two-celled, four-celled cells. And at the end, it reaches to a point where it becomes 16-celled stage. And this is called morula because it resembles a mulberry, maybe like this, I guess. So as you can see here in the zona pellucida is intact, and on day 4 in the uterine cavity these cells get differentiated into an outer cell mass and an inner cell mass. Okay, now something interesting happens. Zona pellucida hatches, and since the inner and outer cell mass is being exposed to the uterine cavity, uterine fluid gets in and creates a blastocele. And this is called the blastocyst, which is getting implanted into the endometrium. So this is our week 1, and now we have our week 2 where we'll be discussing about implantation. Okay, we have our blastocyst here, which has the outer cell mass, which is also called as the trophoblast, and then we have the inner cell mass, which is the embryoblast. And trophoblast is the one which helps in the implantation in endometrium. Like, okay, let me just show you like how it gets implanted. Okay, if we see it in the cross section, okay, this is not a cross section. Okay, sorry, let me draw a cross section real quick. So if we see a cross section of how invagination or implantation happens, we can see like this, and this is okay. So as we can see here, this is how an implantation looks. This light blue invaginations that are here is called a syncytial trophoblast, and the dark blue one is the cytotrophoblast, and above that we have the epiblast and the hypoblast. Okay, this is what is happening in week 2. Now we have week 3 where gastrulation happens. Now if we see a cross-sectional area of the epiblast and hypoblast, we can see them as a bilaminar layer. And in the bilaminar layer, we could see a streak. This is called the primitive streak, and wherein the cells within itself gets migrated towards a nearby layer, and these migratory cells form another layer between the two layers and forms the ectoderm, mesoderm and endoderm. As you can see here, the bilaminar layer becomes a trilaminar structure. Last, we have the neurulation happening, wherein these 3 laminar layers where we see a notochord formation within the mesoderm, and the tubal invagination called the neural tube. This is it for now.